Hello, everyone. I'm very happy to have the opportunity to give you an introduction, a didactic introduction to network neuroscience, and that's the study of neural systems or neural processes and how we can understand them through the lens of network science. So I'm very excited to be part of this SAMC program. We'd like to thank the SAMC program, Data Science and Social and Behavioral Sciences, sponsored by the National Science Foundation through multiple awards. And today what I'd like to do is to focus on understanding or on describing what the field of network neuroscience is, but then also asking the question of where we as a field could go. I think there are many opportunities that I'm hoping to foreshadow for you or outline for you today. So just to get started, a brief outline of the structure of the lecture today is as follows. So number one, we'll go through an introduction to and background to the computational framework, why it makes sense for neuroscience. Number two, we'll talk through case studies of 3 specific applications in the area of neuroscience. Number three, we'll talk about some short descriptions of other uses. We'll then move into model validation methods. And then fifth, we'll talk about a couple options for the future, and then we'll go through a summary of what we've done so far. So I'd like to begin with the introduction and some important background. I think that will help to provide context for the case studies that I'll then describe. So I think in neuroscience, it's absolutely true that there is one fundamental challenge. And that fundamental challenge lies in distilling the rules and mechanisms that have constructed the brain and explain its function and its dynamics, both for humans and for non-human species. And I think efforts to distill those rules and mechanisms have a long history. In fact, we can go back to the age of phrenology, where people believed that the shape of the skull could tell you something about human cognitive processes. So there were particular areas that were thought, whose shape was thought to be related to ideality, for example, causality, human nature, benevolence, veneration, firmness, some very laudable goals, but quite abstract concepts. Now, phrenology has been since replaced by a very formal and scientifically rigorous evaluation of the organization of the brain. And that scientifically rigorous investigation has been made possible by the development of new techniques, experimental techniques and imaging techniques that allow us to see much more than just the shape of the skull. And so for what I'm showing you in the middle here is a picture of a parcellation of the brain into regions that's based on the activation profiles of areas or the structural profiles of areas and other factors from these new imaging techniques. And that has allowed sort of a mapping that in spirit is similar to what people were trying to do in the age of phrenology, but is quite different in the sense that it's actually biologically valid and scientifically justified. In addition to these structural parcellations of the brain into regions, there's also effort to understand functionally which regions of the brain help us to perform particular tasks. So in a magnetic resonance imaging machine, which is present in most hospitals or research institutions around the world, there's the opportunity to understand where regions are active and when. So for example, this is a picture of the brain that has an activation profile of when people are moving their right hand. And what we can do is that we can have people inside of the scanner perform different tasks like move your right hand, move your left hand, speak, be quiet, think about yourself, think about a memory of your grandmother, all sorts of things, millions of different tasks. And from that investigation, we can start to understand which regions of the brain code for or support specific cognitive processes and which regions of the brain code for or support a different cognitive process. So it allows for a generic approach of brain mapping, just like we would map the United States or the European Union or any other large geographic body. We can map the brain in terms of what the functions of specific areas is. So I think this development over historic time periods from phrenology through to structural mapping and functional mapping is great, but I think it's still missing getting us really to where we want to go, which is in distilling rules and mechanisms of brain function. And the reason why I think it's not gotten us to where we want to go completely is that in general, cognitive functions are not explained by the activity of a single brain region. Instead, there's a whole constellation of brain regions and their structural or functional connections that are required to understand a given cognitive function. And that complexity, the fact that we can't be super reductionist, we can't distill it down into a single region that's really driving any particular cognitive function, produces the beautiful complexity in cognition and human behavior that we enjoy as organisms. It also makes the understanding of mental health, neurological conditions, and psychiatric disease much more complicated. But that's an interesting and important challenge. And we'll be just mentioning that a little bit later. Okay, so how do we deal with this fact that the parcellation is not enough? The mapping is not enough. We need to understand circuits. We need to understand the relationships, which regions are involved and their relationships. Well, I think that this is really where network science has a huge role to play in helping us to understand the organization of the brain and how it provides for and supports the kind of cognition that we have. So network science, as you all by now probably know, is a natural language in which to frame complex questions about complex constellations of regions and their connections. So it's an academic field that studies complex networks, from social networks to technological networks to networks in physical systems. And what it does is, in its very simplest form, it tries to separate the complex system into units, which we'll call nodes inside of the network, and then into inter-unit relations, links, connections, interactions, and we'll call those edges inside of the network. And the idea is that this whole pattern of nodes and edges tells you something about how the system really works. That's kind of the underlying assumption. And that's what we are hopeful will be true in the context of the brain. And I'll give you a lot of evidence to suggest that that is the case, but that by distilling this very large complex object into units and their interactions can help us to better understand and make progress on distilling the mechanisms and rules for brain function. Now, I want to say that when we pool the tools of network science over into neuroscience, we have to be careful, and for many reasons, but I think one of the key reasons that we have to be careful is that the network approach is in its essence or fundamentally a modeling approach. So it will necessarily be removing a lot of information. And the open question is, does it remove too much information? Or is it a representation that maintains enough information to help us to understand rules and mechanisms? So when I say that it's a modeling endeavor, I want to be clear about what I mean by that. And what I mean by that is that there are several assumptions that go into this choice of transforming a complex system into a network. And there are at least 3 modeling assumptions that I wanted to raise today. There are more that we could discuss, but I wanted to raise these 3 because they are pretty big assumptions, and they're probably some of the key ones for helping us to evaluate the validity of a network model of the brain. So the first assumption is that you can separate the system into cleanly delineated units. If you can't separate the system into cleanly delineated units, then it's unclear whether we should be really choosing to represent the system with nodes and with edges. The second assumption is that you have to be able to define the most salient relations or connections between units. If you can't define what the edges are, or there isn't a reasonable definition of edges in your system, then clearly network science doesn't seem like the right way to approach the problem. And then third, from the structure of that connectivity pattern that you create, that you encode, that you draw, we can learn something about the system's organization, make educated guesses about its function, and build models of its development, growth, or potentially evolution. So in other words, these 3 assumptions fall into, does the representation of parts into nodes make sense? Does the representation of relations into edges make sense? And then does the network representation really house what you need to understand the system's function? So in order to help you to sort of gain an intuition for when these assumptions, how these assumptions work, I want to talk about a few violations of the assumptions, because I think in the violations you can kind of understand the assumptions more deeply. So the first assumption, that you can separate the system into cleanly delineated units, I think this is an important one because, if we think about the brain, it's a large volume of tissue. And it's in some senses, it's not always segregated in the sense that an acorn is a segregated piece of material. And that multiple acorns might be segregated pieces of material that create the complex system of oak trees and their growth and evolution and dynamics. So I want to ask the question about, for the brain, is this assumption valid? Well, let's think about a system in which that assumption has been violated. And I want to think about ocean currents. So ocean currents are very large movements of bodies of water, and it is difficult to separate those large bodies of water into independent units unless you go from the very, very large scale of currents down to the level of atoms or molecules, in which case, of course, pieces make sense. So I think that in the case of ocean currents, it is often useful to consider using principles of fluid dynamics rather than the tools of network science, or at least some interdigitation between the two. It's unclear whether the pure discrete nature of the network approach is the most relevant for this problem. Now, the second question is, can we define the most salient relations or connections between units? And here, I want you to think about an orchestra. How does an orchestra create music, beautiful sounds? Well, it does so certainly by being composed of pieces. The sort of discrete nature of the orchestra is very clear. So each person in the orchestra, each musician, could be a node inside of the network. And there's a clear delineation between person one and person two. There aren't parts of person two that are also parts of person one. They are discrete units physically in space. But then the question is, what is the most salient relation or connection between these units? That's an interesting question. What is it that two or more musicians share that allow for music to sound the way that it does? Well, you could think perhaps if two different musicians are sharing the same melody, that's an interesting connection or relation that may be important for music. But it's certainly not if every musician was playing the same melody, or if a single line of melody is really all that music was composed of, it wouldn't be this gorgeous thing that we hear when we go to the symphony. So there's also something about harmony, there's something about counterpoint, there's something about foreshadowing melodies in the future or harking back to melodies in the past. There's a lot more complex structure and complex interactions between musical motifs that provide the richness that we hear when we go to the symphony. And because I'm not entirely sure how to encode each of those types of relations into a different kind of edge, I would say that if I was going to try to understand music, I might begin with music theory rather than network science. Now, let's talk about assumption 3. So this one was, from the structure of the connectivity pattern, one can learn about a system's organization, make educated guesses about its function, and build models of its development, growth, or evolution. And here, I want to focus your attention on the bottom left-hand side of this slide, where I have a bunch of words. So words in language are wonderful discrete objects. So we know when the beginning of a word happens, we know when a word ends, and a new word begins. Each word is a discrete object. There aren't half words lying around in our sentences, well, usually. So in language, there are discrete elements, so we're okay on assumption 1. For assumption 2, what are the salient relations between connections or connections between units? In this case, I've linked up several words because they share the same first letter. Okay, so we can question whether that was the most salient relation, but it is a relation. It's a phonetic relation. And so then we can ask, well, from that structure that I've created, can we understand something about language, the function of language? And I would argue that understanding whether words share the first letter is not something that helps us to understand how language functions in general. Language is much more about the meaning of words and perhaps shared meanings in words. And so this particular network that I've constructed does sort of violates assumption number 3. From that structure, we can't learn about the system's organization or make educated guesses about its function. It's been built wrong. It's been built with inappropriate information for the question of understanding language. So I think we need to be careful on all 3 of these parts when we go to the brain, because we want to make sure that we are justified in the delineation of units, that we're justified in the definition of relations, and that from those 2 combined, we are justified in drawing inferences about the system's function. Now, assuming that all of those modeling assumptions are valid in your particular case, then I think the network science approach has a lot of strengths that are particularly useful in understanding the mind and its biological organism, the brain. So the first is that it's a very flexible framework that's applicable to many different data types. And in neuroscience, we have many different data types trying to understand this one system. And it's also useful for testing scientific hypotheses in very different domains, from low scale genetic information up through large scale brain regional information. So as an example, let's think through what are the kinds of questions and scales that network science can approach with the same mathematical formalism in the context of neuroscience? Well, what I've been focusing on so far in the lecture is really this regional level of the brain network, where we can subdivide the brain into parcels or regions, and then we can understand their connections, their functional connections or their structural connections. But that large-scale brain activity provides a sort of coarse-grained encoding of neural processes that are happening at a much smaller scale, so at the cellular level. So we can actually try to understand, if we go down to the cellular level of dynamics, how does that help us to map up to regional dynamics, reflecting the rules of the system's functions? So do cellular processes shape circuit behavior in the brain, and vice versa? How do these 2 spatial and actually temporal scales relate to one another? Well, the nice thing about network science is that we can build a network at the large scale, we can build a network at the small scale, and then we can try to understand how these 2 networks interact with one another to infer causal relations across scales. We can also go even farther down in spatial scale to the molecular level of organization in the brain. We can understand how molecular mechanisms affect large-scale brain structure, and that's very critical for our development of effective pharmacological interventions. So in order for a clinician to prescribe a drug to a patient to help with increasing mental health or to address mental illness, there needs to be an understanding of the organization, how that drug acts on the brain at the molecular scale, and then also how those molecular interactions sort of agglomerate and impact large-scale brain function at the regional level. Now we can also, instead of going down in spatial scale, we can also go up in spatial scale and consider how a single brain network inside one human head is related to brain networks in other human heads. And so that brings us from the level of a single individual to the level of a social group. So this, I think, is really important because there's emerging evidence that social networks offer some external inducers and modulators of our human behavior. So in other words, our social network can modulate how we behave, and how we behave can modulate the social network around us. And so we are not isolated units, and the brain network inside of our head is not an isolated unit. It's actually a connected unit, and it's connected to all of the other individuals inside of our social network. So open questions that the field is trying to answer right now are precisely how do brains shape social networks? And then precisely how do social ties shape the brain? So there's a feed forward and feedback relationship here between the regional network inside of a single human brain and the social network around them. So that shows you sort of the flexibility of the approach across scales. But the other kind of flexibility that network science provides is different sorts of encodings of network models. So in the first few pictures that I've shown you, we've basically been using the simplest network model visualization that you could. And that is where we subdivide the brain into nodes or regions and then edges or their relationships. And in fact, this case in the very middle of this slide is a binary network, so every edge has exactly the same weight. In all of the data I think I will show you today, we will be using networks that have weights on the edges. So they won't be binary, they will be weighted. And so that would be shown pictorially over here in the top left. So this is a network that has weights on the edges, and those weights are encoded visually in the thickness of a line between two regions. But there are many other kinds of encodings that we could consider using and that may be particularly useful for different sorts of scientific questions related to the brain. So one would be to add values to each node. So this kind of a network model is often called an annotated graph. And that's because there's an annotation on each of the nodes. And that annotation can be a linguistic annotation or more what's sort of easier to often use mathematically is a value on each node. So why would we want to put a value on a node? Well, in the human brain, each region has different levels of gene expression, different levels of metabolism, different levels of blood flow, different levels of neurotransmitters, different levels of activity. All of these features of a single region can be encoded inside of the network, and then we can ask questions about how the features of a single region help to explain or help to qualify the architecture that we observe in the connections between regions. There are other extensions that we could consider. So I want to draw your attention to the bottom right here of the slide, which illustrates a dynamic network. So this is where we have a network organization at one time slice, and then another network organization at the next time slice, and the next time slice, and the next time slice. And this dynamic encoding of a network model can be very useful in the human brain for understanding learning or for understanding development as kids grow from very young to young adults, or in late aging when a lot of brain function and brain structure changes as well. So the dynamic networks are important over multiple timescales and can help us to understand evolving processes in human brain organization and function. We can also consider multi-layer approaches. So that is where each layer reflects the organization of the brain in a different way, so, or a different kind of organization. So let's say one layer could show the structural connections between brain regions. Another layer could show the functional connections between brain regions. Another layer could show the patterns of gene co-expression between two regions. So how are two regions showing the same expression of genes? Because that is important for the way that they behave, their activity. So these multi-layer approaches can help us to pull together lots of pieces of information that we have about the brain in a more cohesive way and hopefully provide a more comprehensive picture of what's going on. Now, there are also two other approaches that I'm definitely not going to get into in the lecture today. They would really require another whole lecture, but I wanted to at least mention them. And maybe you've heard of them in some of the other lectures through the SAMC program. So we could use hypergraphs. So that is where there's a set of nodes that are related by sort of a single edge, a hyperedge. And there have been several studies that have used hypergraph approaches for understanding neural systems. So that's definitely something that has been present in the literature, but to a small degree. And I think there's more to do there. And then the second approach that I won't be talking about today is an approach from applied algebraic topology and topological data analysis. And that often encodes the network as a simplicial complex. So as a collection of simplices rather than as a set of dyads. So dyads being connections between a single connection between two nodes. And these approaches allow for I think a richer representation of the data that we can acquire in brain systems. However, we're still in the early stages of applying them to brain networks. And I think that we have more to do in understanding how that richer representation can inform biological interpretations. Okay, so the strengths of the network science approach in the context of the brain are, number one, that it's flexible. And we talked about two ways in which it is flexible. One is that it can be applied across different spatial scales from the molecular up through social networks. And then the second one is that there are different encoding processes that can provide different representations of the system that will be important for different kinds of hypotheses. Now, the second strength that I wanted to mention is that network science provides a rich set of statistical tools, computational algorithms, and theories developed in math, physics, engineering, computer science, statistics, other related fields. And that rich set of statistical tools will be very helpful in understanding what it is that we're seeing in front of us. So this picture that I'm showing you on the right-hand side is a beautiful network. We can tell it's colored, we can tell it has some interesting organization, but what is interesting? How do we define interesting? How do we say that this network is different from some other network? So the statistical tools that network science offers, I think, are a real strength because they help us to quantify similarities and differences in organization. And that in turn helps us to build generative models to really understand why the brain looks the way it does. And to give you a deeper appreciation of the utility of statistical tools, I wanted to mention that there are so many network statistics that have been developed. Each of them have been developed with different goals in mind and often not necessarily with the brain in mind. But I think that that's still a strength in that we have this ensemble of characterizations that we can try to use to understand neural circuits, their relationship to behaviors, their relationship to symptoms of mental illness that are characteristic of psychiatric disorders. And we can also use them to distinguish between groups of subjects, either within a single disorder, let's say schizophrenia, or between multiple disorders. So let's say between schizophrenia and bipolar disorder, or between bipolar disorder and major depression. And I've shown you pictorially a few of these network statistics, and you've probably heard about some of them already through the SAMSI program. So the degree of a node being the number of connections that are coming out of a node, at least in the binary case. The clustering coefficient being related to the number of closed triangles, the proportion of closed triangles inside of the network. Paths being related to the number of edges that must be traversed in order to get from one node to another in the network. Community structure, which is the presence of clustered patterns, clustered nodes that are densely interconnected with one another, but not necessarily densely interconnected with other groups. Cavity structure. So this is something that is easily accessible if you're using the simplicial complex approach that I mentioned on the previous slide. It's much harder to see if you don't use that approach. But cavities become very important for understanding where there are holes in the network, and holes may be indicative of separations in computational processes in the brain, and where that separation is important, perhaps for keeping information isolated for independent processing before bringing it back together again at the other side of the cavity. Hubness is the notion that there are some regions, some nodes inside of a network that are much more interconnected than others. And they can be highly influential nodes in the networks because they can shuttle information to so many other areas. So that becomes important for understanding information transmission in the brain. The presence of shortcuts across the network is also very important for information transmission. So if it's possible to get information from one side of the network to the other through one edge, that can provide swift information transmission, but also transmission that doesn't have as much potential for being altered by surrounding processes. And then core periphery organization being a density of connections inside of a core, and then a sparsity of connections in a periphery, where there's a tendency for peripheral nodes to connect to the core, but not to other peripheral nodes. And I wanted to focus just briefly on these two that I had shown at the top middle, the clustering coefficient and the path length, because I want to mention that the statistics are not just useful in characterization of network organization, but also in building generative models of networks. So why is it that the brain looks the way it does? Well, perhaps if we could write down a simple rule that says, oh, the brain should have this structure and this structure so that it can perform information processing in this way, that would help us to better understand what the brain does and how it does what it does. And such a generative model could be then tested and validated against experimental data and further refined. So some statistics, including the clustering coefficient and the path length, can be used to probe local structure, like the clustering coefficient probes local structure in the sense that it is assessing the prevalence of these triangles inside of the network, but also global structure. And I think that the path length is something that is a measurement of global structure because it takes into account the entire network and asks what is the shortest path to get me from one node in the network to another, if we're talking about the average shortest path. And we can use those two to build generative models of networks under certain assumptions. I'm going to give you one example of how that might work, and then we're going to test it against the brain. So the idea that the clustering coefficient and the path length may be useful markers for a generative model of complex systems has been around for a while. And in fact, I'd like to highlight the work of Watson-Strogatz in their 1998 Nature article, where they build a simple model of constructing what's called a small world network. And small world networks have this wonderful property of relatively high clustering, which allows kind of local segregation of information in a system, as well as, on average, very short path length. So you can get from one piece of the network to another piece of the network with few hops. And that's possible because of these long distance connections that you can see inside of this ring lattice network. Now, what Watson-Strogatz did is that they showed that you could begin with a regular network. Here's a regular ring lattice where every node is connected to its k nearest neighbors. And then you can start rewiring that network with some probability, starting at p equals 0 and then up through p equals 1. And as you go from the regular lattice through this rewiring to a random graph, you move through a regime of small worldness where you have very, very high clustering coefficient and on average, very short path length. That small world this is a generative model of small world organization. And the short path length, the high clustering coefficient and short path length provides this very interesting balance between the potential for local information processing and global information transmission. And that is something that we think is very important for a brain and actually for many other kinds of information processing systems. But the clustering and the path length are not the only statistics that can be useful for building generative models of brain networks, as we're going to see later. So we will definitely talk about community structure and some of these other ideas as well. I just wanted to highlight clustering and path length because that harks back to some of the important historical work that has motivated a lot of open questions in network neuroscience. So that brings me to the case studies. In the first case study, we're actually going to test that small world model of the generative model of the brain. But I'll also talk through two other case studies that hopefully together will help give you a flavor of the kinds of questions that are being asked in the field. So in the first case study, we're going to talk about modeling structure. Modeling the structure of the brain. And for this part of the talk, we'll be focusing largely on structural connections between brain regions. I want you to look at this picture here on the left-hand side. This picture it reflects data that can be acquired using a magnetic resonance imaging machine. And an MRI machine can take images of the diffusion of water molecules inside of the brain. You may not all know that inside of your brain, there are water molecules bouncing around continuously. They're moving by Brownian motion, and they are continuously bumping up against structural boundaries. And these structural boundaries are actually long tails of axons between one neural cell body and another neural cell body. And so that long axonal tail is the track or the highway that allows for information to be propagated from one information processing unit, a cell body, to another information processing unit, another cell body. And so what scientists have realized quite recently is that we can study the diffusion of water molecules to infer where these tracks are, because they are boundaries along which the water molecules bounce. And each of the threads that you are seeing in the bottom left figure are big bundles of these axonal tracks that connect up one piece of the brain to another. So in other words, we can gather information about the precise wiring, the precise pattern of highways inside of each person's brain. And importantly, this kind of imaging can occur when somebody is happy, healthy, awake, behaving, just inside of an MRI scanner. So there's no need to wait until a person passes away to do ex vivo work. This is something that is in vivo, there is no adverse reactions to this particular imaging technique. It's something that is just, it's a beautiful window into what the structure of the brain really looks like. So this is the structural network organization inside your head, and we want to understand how that structure is organized. So we'll talk about modeling structure in the first case study. In the second case study, we'll move from structure to function and talk particularly about modularity, which I mentioned before is something that's going to be important. And then after talking about modularity and function, we'll move out from the brain to sort of the world. So we'll start sort of very internally focused, and then we'll move out and ask, how does the mind build models of the world around us? And it turns out that the world around us has its own network structure. And so what the mind has to do is actually to learn a network. It has to use the network inside of the mind to learn the network in the world outside. And so there's this interdigitation between two networks, internal and external. How does that all happen? So we'll talk about how the human mind, what we know so far about how the human mind models the network organization of the world around it. So let's start with the first case study, and that is modeling structure. So we can begin with the human, which I'm showing you over here on the right-hand side, and we can use this diffusion imaging approach that allows you to map out the diffusion of water molecules and where they're bouncing against the boundaries of these large axonal tracks that connect a set of cell bodies to another set of cell bodies. So we can study that, but we can also use similar kinds of imaging approaches in macaque monkeys, in Drosophila, which is a fly, and in the mouse. And so this is work that was read by Rick Betzel when he was a postdoc in the lab. He's now a faculty member at Indiana University. And what he did is that he took structural connectivity patterns from each of these different species and asked, are there consistent principles of wiring present in all of these? And if there are consistent principles of wiring, what are they? And can those, if we can understand what those principles are, can we build generative models of how the networks came to be this way? So he began with a pretty simple analysis, which was to take each of these connections inside each of the animals and ask, what is the distance of the connection? How long is it physically? And then what is the weight of the connection? How strong is it? And in the context of diffusion imaging, it's related to how many connections are estimated to be going from one region of the brain to another region of the brain. So what I want you to notice is that across all of these different species, there's a negative relationship between the weight of an edge and the distance of the edge. So in other words, most connections are actually quite short, and then a few connections are long. But importantly, also the relatively short connections are the strongest, and the longest connections are the weakest. So this is important, I think, because this negative relationship between weight and distance is somewhat reflective of what we would anticipate in a weighted small world model of the brain. So remember, we had talked about this small world model where there's a preference for local connections that allow for this high degree of clustering, and then a few long distance connections. That preference for local connections, but the presence of a few long distance connections, is also what we see very consistently across each of these species, from mouse to fly to the macaque monkey and then to humans. But importantly, if the small world organization and this particular generative model is relevant for neural systems, then we should test some of its explicit predictions. One of the predictions of this model is that long distance connections in these systems should be placed at random. And why do I say that? I say that because the way this model works is that the local connections are present early, and then the long distance connections arise when a current edge is randomly placed in a different spot in the network. So there's a random rewiring of an edge. And because it's a random rewiring that gets us into this small world regime, the assumption is that these, or the prediction / assumption of the model is that these long distance connections are placed at random in the network. So what I want to do is to go back to experimental data in neuroscience and ask, are long distance connections placed at random, or are they highly non-random? If they are highly non-random, then that would suggest to us that the small world model is not a sufficient explanation for the organization of the human brain or the mouse brain or the macaque brain. All right, so let's look at the data. So we're going to go through those same species again, and we're going to ask the question, are long distance connections placed at random, or is there some shared structure between where the long distance connections are placed? So to do that, I want to quantify the similarity between node I's connectivity profile to the rest of the brain and node J's connectivity profile to the rest of the brain. So we have two nodes, node I and node J. I'm going to ask how similar are their connectivity patterns to the rest of the brain. And we're just going to use a simple measure of cosine similarity for that difference, or for that similarity. So then we can say, well, if we just focus on the very long distance connections, is there high cosine similarity? So this node tends to connect to the same ones that this node does, or is there low cosine similarity? If long distance edges were placed at random, we would expect low cosine similarity. If long distance connections are placed with some shared principles, then we would expect high cosine similarity. Okay, so let's look at the data and see what happens. On the left-hand side here, you see the y-axis is the mean cosine similarity. Along the x-axis is the percentile of long-distance connections, with the first bin being the 5% longest connections, the second bin being the 10% longest connections, the third bin being the 20% longest connections, and the fourth bin being the 25% longest connections. In the colored bars, you see the true cosine similarity for that species. In the gray bars, you see the expected cosine similarity if the network has been rewired at random. And so what I want you to notice is that the colored bars are consistently higher than the gray bars. And what that means is that the cosine similarity of these connectivity patterns, particularly for the long distance edges, are higher than expected in a randomly rewired network. That means that long distance connections are not placed at random. There's some shared variance here that's driving that cosine similarity up higher. And what that also means is that the small world model is not going to be a sufficient explanation for what we're seeing. So I just walked you through the mouse, but I just want you to look at the Drosophila as well. Notice the higher green bars than the gray bars, same with the macaque. Same with the human at low resolution imaging and at high resolution imaging. You can particularly, the most striking data is here from the human, where the cosine similarity in the true data is so much higher than in the randomly rewired network. All right, well, so if the small world model is, predicting that those long distance connections should be placed at random, but we see from the data that the long distance connections are not placed at random, then we have to go back to the drawing board. What is a second candidate generative model that would help us to explain the connectivity patterns that we observe? So if we think about it and if we go back into the literature, there have been several other candidate rules proposed by investigators for why long distance connections are placed the way they are and where they are placed. So we definitely know, for example, that the brain is characterized by local modules. And we also know that those local modules are connected. They're not completely disconnected. They're connected by long distance connections. So one hypothesis in the literature is that long distance connections are purposely placed to connect two modules together. Now I want to walk you through this visualization because it's going to be important in the next couple slides. So I want to make sure that you understand what you're seeing. So on the left-hand side is a picture of the graph itself that we'll be studying. So every node is a region in the network, and then every edge is a connection in the network. Here you're seeing that there's clear modular structure in this particular graph. So there's a density of connections inside of a module, and there's a sparsity of connections between modules. The connections inside of modules are short physically, and the connections between modules are long physically. Now on the right-hand side is an adjacency matrix representation of the same graph. So the data on the left is the same as the data on the right. It's just encoded differently for visualization purposes. So each of these 4 different modules can be seen by these blocks along the diagonal, blocks of dense connections along the diagonal. So one particular block reflects the connectivity in one particular module. Then if you look at all of these off-diagonal blocks, you can see a few connections randomly interspersed in here, and those are these long-distance connections that are placed between modules. Okay, so hopefully that helps you to understand. The axes here should be nodes by nodes. And the color of the edge indicates its strength. So we can definitely look for this kind of structure in brain and networks using community detection algorithms. We often use modularity maximization as the community detection algorithm of preference in many brain network studies, but there are also other techniques that are available. We've used InfoMap before, and there are spectral clustering approaches and others. I'm sure that you'll be learning all about that in some of the other lectures during this SAMC period. So modularity can be detected. We can figure out where the modules are and whether those long distance links are placed between modules. But before, and actually even without doing that, I want to take a step further and say, is modularity enough to explain the kinds of information processing needs that a brain may have? And I don't think it's enough. And let me tell you why. So I think that there are distinct architectures that a brain network has to have in order to facilitate very diverse information processes. On the left-hand side, you're seeing that modularity picture again, where you can see modular organization along with these blocks along the diagonal here. And then you can see some sparse connectivity between the modules. That structure allows for information integration inside of the modules, or between modules, and then segregation within a module. So that's definitely important, but I want you to also consider 2 other architectures that may be important. Bipartite structures, where we have connectivity preferentially between 2 groups of nodes rather than within groups of nodes, could be very helpful for targeted information transmission rather than processing. And that is illustrated on the left-hand side by the graph, but I also want you to notice the structure on the right-hand side in the adjacency matrix. So bipartite structures are obvious by the density in these off-diagonal blocks rather than the on-diagonal blocks. So this block, for example, says that module 1 is heavily connected to module 3 and vice versa, but the module 1 and module 3 themselves are not particularly intra-connected. So that's the bipartite structure that I think is important for information transmission. But we can also think about core periphery structures as being important for information transmission too, and specifically for information broadcast and receipt. So on the left-hand side, I'm showing you the visualization of the graph where you can identify the core in the bottom left. So this is a set of nodes that are densely intra-connected, and they also have connectivity to the 3 other modules, allowing a broadcast of information and also a receipt of information back into the core. These 3 external modules are peripheral in the sense that they are not particularly well connected internally, but they are very strongly connected back to the core. And the adjacency matrix that would reflect this kind of organization is shown on the right-hand side, where you can see the core in the top left and its connectivity to the other modules along the top of the adjacency matrix and the side of the adjacency matrix. And then you can see the sparsity of connections among peripheral nodes in the dark blue lower right block of the adjacency matrix. So I think that there's certainly evidence in the literature that neural systems and actually other information processing systems might need to use all three of these different architectures to support different information functions. And what we find, and this is again work that was led by Rick Betzel in the lab when he was in the lab, is that brains display these distinct architectures superimposed upon one another. So in other words, the brain is not particularly well characterized simply by modules that are densely intra-connected. It also has evidence for bipartite structures and for core periphery structures. And it's the combination of modularity, core periphery structure, and bipartite structure that best encapsulates the organization that we observe in brains. And I think that that's really neat because it is supportive of the fact that the brain has many diverse information processing requirements, and that diversity is best supported by diverse structural motifs, from modularity to core periphery structures to bipartite structures. So how can we determine that this is a good model for the brain? I sort of claimed it, but I want to ask, how would we determine that? Well, we can use what's called a stochastic block model, and we actually use a weighted stochastic block model because our edges are weighted. We fit that stochastic block model to the adjacency matrix, and that allows us to determine where these white lines should be placed that demarcate the separation between one block and another block. And I'm not going to get into the math of the stochastic block model, but I'm just going to sort of talk about the intuitions. You can always look it up if you're curious for more information, or seek out other online lectures on the topic. So the stochastic block model is searching for a way of subdividing the adjacency matrix into blocks such that the probability of connectivity inside of a block is relatively homogeneous. And so that helps us to determine where modules are, where periphery regions are, where bipartite structures are. And if we take that stochastic block model, weighted stochastic block model, apply it to actual data, we can determine where the boundaries are. And then we can ask questions about what those boundaries help us to understand. So in these next few slides, what I want to do is to ask, can we use the weighted stochastic block model to better understand the brain's function? In other words, let's take this computational approach, apply it to brain data, and ask what it tells us about how the brain works. So the first, I'm going to give you two examples of answers to that question. The first example is in the context of the mouse connectome. So in the neuroscience literature, the word connectome is often used as to mean the brain network inside of an animal. So the mouse connectome, or the brain network inside of a mouse, can be studied using the weighted stochastic block model. And what I'm showing you here along the X and Y axes are different terms for regions of the mouse brain. Olf standing for olfactory areas, hip standing for the hippocampus, auditory, odd standing for auditory regions, vis standing for visual regions. This set of areas are important for sensation and motion, for example. And what we can do is that we can apply the weighted stochastic block model to the connectome, and that provides us with these white lines, which is the demarcation between different blocks in the connectivity architecture, where the connectivity inside of a block is relatively homogeneous. Now we can ask the question, I'm not showing you the connectome data at all, I'm just showing you the white lines so that you can see where they are. And then I can say, well, does that organization tell us something about the brain's function? And one particularly important marker of brain function is patterns of gene co-expression. So when two regions of the brain share the same expression of genes, that's often a marker for shared function. And so here, what you can see is the same white lines that I've shown you on the left-hand side, which were derived from applying the weighted stochastic block model to the structural connections of the mouse. And on the right-hand side, I'm coloring each region by their amount of gene co-expression. And what I want you to notice is that even though these white lines, these demarcations, were defined based on structure, they provide a really beautiful set of boundaries that also demarcate areas of shared gene expression. So that suggests to us that these structural principles are explaining patterns of function. So here, just to walk you through this a little bit more, you can see that there is a set of areas that have very high shared gene co-expression, and they are also predicted to be in the same block. You can also see in darker blue sets of regions that have very low shared gene expression, and they're predicted to be in a separate block. And so I think that the fact that these white lines provide very clean demarcations between areas of shared gene expression is indicative of the fact that the structure of the brain network informs or predicts or explains the function of the brain network. So that's sort of the first answer that I wanted to provide. And then the second answer is moving from the mouse to the human. And here what I want to do is to take the same approach. I'm going to show you regions of the brain along the x-axis and then the y-axis. And I'm going to show you the white lines that were provided by applying the weighted stochastic block model to the diffusion imaging data. So that's where we found the highways or axonal projections between different regions. And now on the right-hand side, I'm going to overlay patterns of function. And in this case, instead of gene expression, I'm going to show shared patterns of brain activity. So here are shared patterns of brain activity where brighter yellow colors indicate more shared activity. And this is simply a Pearson correlation between regional time series of activity. And then in the lighter blue, there's going to be that indicates a negative relationship between time series. So again, what I just want you to notice by briefly looking at this is that the demarcations of blocks from structure are highly explanatory of patterns of shared function. And so that suggests to us that not only is the stochastic block model a better generative model for the brain organization going beyond the small world model, but also it helps us to better understand the function of the brain, both in terms of gene expression and in terms of shared activity. So just to quickly provide you a summary of case study 1, we started by saying that network statistics can be used to model the structure of the brain with increasing precision. And then we moved into structural models that can be tested against experimental data and increasingly refined. So we noticed that the small world model, when we tried to test it against experimental data, fell short in predicting that the long distance connections should be placed at random, when in fact, they're not placed at random. There's a lot of shared variance as indicated by the high cosine similarity. Then we talked about structural models, particularly the weighted stochastic block model, as one that allows us to understand why brain function and gene expression are organized in the way that they are. So that allows us to move on to case study 2, where we're going to span from structure to function. And we're going to talk a little bit about modularity. So in this particular case study, we want to understand how the architecture of structural and functional circuitry underlie cognitive functions and also cognitive deficits in disorders of mental health. And before we get into the details, I wanted to walk you through this really neat visualization that Rick Betzel created while he was in the lab. So on the right-hand side, you see the brain, and this is the right hemisphere. So on the right-hand side of your body, and it's on the outside, what you're seeing. And then this is the medial surface. So if you were able to look at the very center of the brain and see inside, this is what you would see. And then here's the left hemisphere, the outside part. And then this is the left hemisphere, the inside part. So from this, and then, I should say that each region is being color coded according to which module that region belongs to. So, and then on the right-hand side, you can see the actual data from which we determine what the modules are. So here, every region is a node inside of the network, and shared activity. So when two activity time series are highly correlated with one another is shown by an edge. And we've thresholded the graph simply for visualization purposes, although all of the data that we use is fully weighted. So what after we did that, we applied a modularity maximization approach to identify where the modules are in the brain. So where are the sets of regions that are densely connected versus the sets of regions that are less densely connected. And the color over here maps onto color on the right-hand side. So you can see here that the blue module is this visual regions back here in the very back of the brain. Those are areas that support your ability to process visual information. But there are other regions of the brain that are important for motion. So those are these cyan and orange regions here, which you can see are this module over here. And then there are also regions of the brain that are important in self-reflection and self-referential processing and things like mind wandering. And those are these red areas here. They have a particular name. They're called the default mode system, and they're present in a little bit in the back of the brain above the visual areas, a few sections over here in the frontal cortex and temporal cortex, and then some regions along the midline, precuneus and medial frontal areas. Don't worry about the names. You can remember that there's a module in your brain that's important for mind wandering. That's probably more relevant for you right now. Okay, so from this picture, I hope you can tell that modularity is a very clear organizing principle for functional connectivity or for a functional network of the brain. And you can see that by eye just by looking at the picture on the left-hand side. Okay, so we want to go a little bit beyond understanding that pattern of functional connectivity and ask, what's it useful for? Why would we have a very modular structure inside of our brain? And when we're thinking about answering that question, we can go back into the literature and ask, what have other areas of biology seen in terms of ways that modularity is useful for an organism? And we can certainly go back to a long history of work in evolution and development and in adaptation that has underscored the fact that modularity of structure, modularity of morphology, modularity of function is important for a system to be evolvable. And so we can even go back to, let's say, Darwin finches and notice that the different pieces of the beak or the different sections of the skull or the different parts of the organism of a bird, the fact that they are parcellated, that there are pieces or there are modules, allows for there to be developmental or evolutionary programs that change one module without adversely affecting other modules in the system. So modularity allows for evolution and development. There's also some very clear work more recently in neuroscience to suggest that there is a very precise modularity of mind where there are cognitive processes like mind wandering that are separate from other cognitive processes, and there's not a continuous deformation of one process that will lead you to another. There's sort of a demarcation between processes. One could think about, is there a continuous deformation of auditory processing that would create visual processing? The argument in the literature is no. These are quite different and different enough that we call them separate modules of processing. But we want to go to the architecture of the brain network and ask, well, is there modularity in the brain network? And if there is, does that help to explain not just the differences between cognitive functions, but also their adaptation, their evolution, their development, their change? And that becomes particularly important when we try to understand human learning. In order for a human to learn, to change their behavior, to change their mind, to change their skill set, there has to be a level of adaptation. And we can think about adaptation as a short time scale correlate of long time scale adaptation through evolution and development. So since other areas of biology have shown that modularity is important for long time scale adaptation, we can hypothesize that modularity of brain networks is important for short time scale adaptation in the context of learning. So this discussion brings us to two questions. One is, are brain networks modular? And then two is, does modularity help us to understand the large-scale signatures of adaptation and learning in humans? And there are a couple theoretical challenges and computational challenges that we would have to address and that have been recently addressed in the field that allow for us to ask these kinds of questions about human brain networks. The first one is the challenge of parsimoniously representing and describing complex connectivity patterns. And as we've seen through the last case study and through the introduction, we can do that with network models. And that's kind of a new paradigm, a new perspective in the field of neuroscience, that we can represent the whole brain, not just study single regions, but represent the whole brain as a pattern of connectivity and understand its organization. The second challenge is to detect modular structure in models of brain connectivity. And modularity maximization is one that we have been using. It was applied in the context of neuroscience, I think first by David Monnier, who was at the time a postdoc in Ed Bullmore's lab at the University of Cambridge. This is a paper that was published in 2009 in NeuroImage, which I think was the first application of modularity maximization to brain networks. And then after addressing that challenge, we also need the ability to detect evolving modules. So if we want to understand adaptation, learning, change, we need not just one snapshot, which is what I showed you on the first slide of this case study, but we need a way of tracking how those modules might be changing, just like the Darwin Finch beaks are changing, right, over evolutionary timescales. So for that, we use multilayer modularity maximization, and we use particularly the formalism that was proposed by Muka et al. In their 2010 paper in Science. And I'm not going to go into the details of these approaches. Again, I'm going to stick pretty high up at the conceptual level, but I have placed references throughout that you can go look up if you're interested in understanding the details more precisely. Okay, so by addressing each of those challenges, we can now ask the question of whether temporally changing modularity happens in the brain and whether it's related or helpful for us to understand adaptation and learning. And so here is one of the studies that I wanted to highlight that begins to address that question. And it studies what's called sequence learning, which is a sequence is a pattern of finger movements, similar to a piano arpeggio, or it doesn't have to be piano, it could be violin. I don't play violin, so that probably doesn't look right. But so it could be a pattern of finger movements, could be typing on a keyboard, could be on a musical instrument that creates a sequence. And in this particular study, we had sequences that were 12 notes long, and people practiced them over and over and over and over and over again for 6 weeks. And we brought them into an MRI scanner on the first day of training, and then 2 weeks later, so at 2 weeks, 2 weeks later at 4 weeks, and then again finally at 6 weeks. So we had images taken of their brain while they were practicing it 4 different times, but then they practiced in between those daily at their own computer. So there was striking amounts of learning. And when we looked at the brain, we found that there was this really interesting modular structure that changed as people were learning, consistent with the idea that modularity may be important for adaptation and learning. So on the bottom, I want you to focus on the bottom here first for an intuitive visual, and then we'll go into the statistics at the top. So at the bottom here, you see a matrix that has brain regions along the y-axis and brain regions along the x-axis. And these regions are taken from the motor cortex, which is shown over here on the brain in green, and the visual cortex, which is shown over here on the brain in yellow. The motor and the visual cortices are particularly important for finger sequence learning because you need to be able to move your fingers, so that requires motor cortex activation, but you also need to be able to look at the screen to determine which are the movements that you need to be playing next. And so that requires the visual circuit to be involved, because the visual circuit takes the information from the screen and then transfers that information into your fingers for your motor response. So here you can see in the first adjacency matrix on the left-hand bottom left, that there's a strong block of connectivity among visual areas. That's this large red block at the top left. And there's also strong connectivity among motor regions, but there's not particularly strong connectivity in the off-diagonal blocks, which would be connections from a visual region to a motor region or vice versa. So the block diagonal structure indicates clear modularity, and the weakness in the off diagonal structure indicates that these two modules are slightly untethered from one another. Now, this was the image that we took at 0 weeks of practice on our first day. The next image is what happened at 2 weeks of practice, and then 4 weeks of practice, and 6 weeks of practice. So I want you to look across the bottom of the slide and realize that these off-diagonal blocks move from yellowish green to lighter blue, to lighter or darker blue, to very dark blue. And what that means, the color here is indicating the allegiance of regions to modules. And what you can see is that the allegiance between the visual and the motor system is becoming less and less and less. So in other words, these two systems are becoming more and more independent, autonomous as learning is happening. And to see the statistics of that, you can look at the top. So here is motor visual integration. That's how much connectivity there is between the motor and the visual modules as a function of the number of trials practiced, which is along the x-axis. And you can see a clear negative relationship here. The markers indicate the average over subjects, and then the error bars indicate the standard error of the mean across subjects. If we just take the marker values themselves, the correlation here is an R of minus 0.93. So very, 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 very strong negative correlation between motor visual integration and the number of trials practiced. So again, suggesting that as learning happens, the modules in the brain, which initially had to come together to enable learning to happen, become more and more autonomous as the performance of the behavior becomes more autonomous, more of a habit, more ingrained in motor memory. So we followed that study up with some additional studies to evaluate how much these modules are flexible, not just in the context of sequence learning, which is one particular example, but in terms of lots of other kinds of learning, as well as what's called executive functioning, which is the human capacity for working memory, for making difficult decisions, for inhibiting inappropriate behavior, and many other kinds of functions that are very strongly present in humans, sometimes less strongly present in other animals, and certainly something that develops a lot during our lifetime and has not really reached adult levels until about the age of 25. So what we found is that flexibility in network modules, so the ability for modules to, regions of a module to sort of communicate with another module and then come back or change which module they're communicating with, predicts individual differences in visual motor learning. It can also predict future learning. So how flexible a brain is on the first day of practice can predict learning 6 weeks into the future. And it can also predict how swiftly you learn, so the actual learning rate. The flexibility of modules inside of the brain is also related to a person's cognitive flexibility, also related to their working memory, their planning and reasoning. And it's also an intermediate phenotype for schizophrenia, meaning that it is lower in people who have schizophrenia, higher in people who are healthy, and sort of intermediate in relatives of people who have schizophrenia. It can be modulated by medication, which is very interesting, and so it may be something that could be targeted with pharmacological interventions. And we also find that it is related to mood. So people who are in a more positive mood tend to have greater flexibility in their modular structure than days when someone is in a less positive mood. So this is, I think, really an exciting direction for the field, which is to ask questions about how, not just whether the brain is modular, but how it's modular. And the answer is it's flexibly modular. And then start to ask questions about why is it flexibly modular, and how does that flexibility allow for a balance between the performance of specific cognitive processes and then the necessary interdigitation of cognitive processes when we have to learn, or when we have to respond, when we have to plan, when we have to reason, when we have to be flexible cognitively between different rules or paradigms in our behaviors, etc. There's also this beautiful review article if you're curious about plasticity following injury, brain injury or stroke or other events. There's a beautiful paper that suggests that flexible modularity is what helps us to plastically revise brain network organization following interventions. So just as a quick summary of case study 2, we found that not all network statistics may be relevant for understanding the mechanisms or emergent properties of brain systems. We went back into history and found that modularity seems to be something that's been noted as important across many areas of biology in the context of evolution and development. And so we focused on modularity because of that special relevance. And we focused on taking that notion that modularity is important for adaptation, bringing it down to the small temporal scales of learning. We found that brain networks were modular and that they were flexibly modular, and that flexibility in the modular organization supports many aspects of human cognition as well as learning, and can in fact even predict learning in the future. So that brings us to, I think, a really nice place to stop the first part of this lecture. So you can get up, stretch your legs, and we'll come back for the second part of the lecture in a few minutes.